That's that. All right. Let's see. 1, 2, 3, 4, 5, 6, 7 missing. That's not bad. Okay, if you haven't signed into attendance, please do that. Give it a couple more minutes, and we'll get going at 9:05 or so. Any questions from Friday regarding what we covered in that Z diagram? Okay, doesn't look like it. All right then. Oops, wrong program. Okay, 24, that's not bad. Okay, so we did a Jamboard on this thing yesterday, or the other day, where I basically walked you through how the light energy of the sun gets converted to chemical. So at this point, what are the two main products of the light reaction that the energy of the sun has been converted to? So what's the chemical, where's the chemical energy stored at the end of the light reaction? Yeah, the ATP, which is down here, turn on my laser, and the NADPH, which is over here. So those two are going to go on and make sugars later. But let's look at how this chemiosmosis actually works. Okay, so this is the electron transport chain of photosynthesis. It should look somewhat familiar. What's it look like? Electron transport chain in mitochondria. Okay, so similar to the mitochondria, you've got protein complexes that are embedded into the membrane. You've got an ATP synthase. You've got a hydrogen ion gradient, and you got electron transport chain. Okay. All right. One thing that's different, where is the higher concentration of hydrogen in a chloroplast? The inside of the membrane or the outside of the inner membrane? Yeah, right? And note the orientation of the ATP synthase. It's the other way around. The ball is sticking out to the stroma, whereas in mitochondria, the ball would have been in on this side. So those are two small differences. Okay? So we've got this complex, right, which is photosystem 2. Yeah, it's not. Okay. Sunlight's going to come, okay, and hit a foot of the chlorophyll molecule, okay, and they're going to start going down this electron transport chain. You have to replace those two electrons. So there's going to be a Z protein at one here, which is going to split water, right, and donate 2 electrons back to photosystem 2. The electrons then, and oxygen will be given off. Okay. The electrons then will go through this system. As the electrons pass through this cytochrome complex, 2 hydrogens will be pumped into the interior. Okay. That's going to form a hydrogen gradient. That hydrogen gradient is going to go out through ATP synthase and make ATP. So that's step 2. Okay. The electrons keep going. When they get to photosystem 1, they'll replace the electrons that were lost previously, and then light will hit them again, and 2 more electrons will get knocked off and continue on their way. They're going to flow through until we get to NADP plus reductase. And here, we're going to store the energy as NADPH. Okay? Know this diagram because there is often a compare and contrast mitochondria and chlorophyll ATP synthase or chemiosmosis or electron transport chain question on tests. Okay? So be sure that you can give similarities and differences, compare and contrast between between how a chloroplast works and how a mitochondria works. Okay? And then the two substances, NADPH and ATP, go on to the Calvin cycle where they're actually going to start making the sugars. Okay, so looking at the electron transport chain. Okay, the electron transport chain uses light energy from the sun to produce the following. Okay, ATP and NADPH. They're going to go off to the Calvin cycle. Photosystem 2 absorbs light. Okay, and the excited electron passes from chlorophyll to a primary electron acceptor. That's that excited molecule, and then the electrons keep going. We need to replace the electrons that were lost in PS2. So we're going to use an enzyme to split water and take the electrons from the water and give them to photosystem 2. Okay. The oxygen from the split water will combine with another oxygen from a second split water to form O2. Okay. That oxygen is then released into the atmosphere. Okay. And then we use it in cellular respiration. Okay, now, going back to that Z diagram. Here, okay? Electrons enter here from water, and they leave here in NADPH, okay? So in this system, electrons come in at one point, and they leave at the other end. Because of that, okay, the system where the electrons come in from water and leave as an NADPH is called non-cyclic photophosphorylation. Non-cyclic because electrons come in at one point, leave at the other end. So they come in from water and they leave as NADPH. It needs light, so photo, right? And then phosphorylation because we're making ATP. Okay, so that's what this process, this Z diagram, is called, where you have electrons coming in from one end and leaving the other. Okay, so the light reactions excite or elevate the electrons in two steps, PS2 and PS1. Okay, PS2 generates the energy as ATP. PS1 produces reducing power or reduced molecule NADPH, right? That's basically the difference between 2 and 1. 2 makes ATP and 1 makes NADPH. Okay? So there's the two molecules that we've made. Okay? But there is a second way that this system can work. It's called cyclic photophosphorylation. Okay? The difference is this word here, cyclic. Okay? So in cyclic photophosphorylation, electrons don't come in from one end and leave the other. The electrons instead cycle through. They loop. Okay? If for some reason, and I haven't gotten into the reason yet, but if for some reason NADPH can't be made, then the electrons will go back to PS2 and then come through the system again. So in that case, you're just going to make ATP, but you don't make any NADPH. Okay? And I'll explain why this happens in a minute. I'll give the explanation first, then I'll tell you why. What this does is it coordinates the sugar synthesis process with the energy available from the light reactions. Okay, so it's a coordination step. Okay, it turns out Calvin cycle uses more ATP to make glucose than NADPH. Okay, the ratio is actually, come on, you need 18 ATP to make a glucose, but you only need 12 NADPH. So you're going to run out of ATP first because you need more of them. Okay. When the system starts running out of ATP, the light reaction will switch from non-cyclic to cyclic. So the electrons will simply loop back here and they'll do this until you have enough ATP to make more glucose, at which point the system goes back to non-cyclic. Okay? So not cyclic photophosphorylation is a way of making sure that you have enough ATP to make the sugar. Okay? So what 18 ATP and 12 NADPHs make one glucose molecule. Okay? So in photophosphorylation, right, non-cyclic means energy comes in this side, electrons come in from water, pass through the system, and make an ADPH. Okay, so it's that pathway for non-cyclic. Okay, so you get ATP and an ADPH both. But in cyclic photophosphorylation, you don't make any NADP. The electrons come in up to the end of photosystem 1, and then they cycle back to make energy, more ATP. So this part of the pathway isn't used. Okay? So you still generate ATP, but no NADPH. Here's a question. If the cell makes excess ATP, what's the plant cell gonna do with that excess ATP? It could do that, but it's just ATP, right? The cell can use it for anything it wants. Any power requiring process in the cell, it'll just take that excess ATP and use it for whatever it needs to use it for. It can dump it into synthesis, it can dump it into chemical reactions, do whatever it needs to. It's not just splitting of water. It's just ATP, right? So that's another way of using up the excess ATP if it makes too much using this process. Okay, so where did the energy come from? Okay, where did the electrons come from? Where did the water come from? Okay, but before that, yeah, cellular respiration. Where's the oxygen coming from? What process of photosynthesis? Yeah, splitting of water. Where's the oxygen go? Into the air. Where did the hydrogen ions come from? Water again. Where did the ATP come from? Photosystem 2. What's the ATP going to be used for? Right? To make sugars. Where did the NADPH come from? PS1. And what will it be used for? Also Calvin. Okay? So you should be able to answer all these questions in order to know how the light reactions work in terms of where products and reactants are going. Okay? So stay tuned next for the Calvin cycle. Any questions on that? Okay, let me post Calvin cycle up for this week. Actually, let me see if there's any, before we do that. Which one's a better video to show you guys? No, not that one. I'm going to show you the other one. Okay, so present, tab. Okay.
Plant have cells that carry out photosynthesis. If we zoom in on this photosynthetic plant cell, we can see the chloroplast. The leaves of this plant have cells that carry out photosynthesis. Zoom in on this photosynthetic plant cell, we can see the chloroplast, where the reactions of photosynthesis occur. Photosynthesis consists of two primary steps, the light reactions and the Calvin cycle reactions. In this tutorial, we'll focus exclusively on the light reactions. The light reactions occur within the thylakoid of the chloroplast. Here, special pigments absorb light energy and transfer it to high-energy electrons, eventually producing ATP and the electron carrier NADPH. Let's zoom into the thylakoids to take a closer look at how ATP is created. The light reactions use two photosystems, called photosystem 1 and photosystem 2, which are both embedded in the thylakoid membrane. It's important to realize that these photosystems are named for the order in which they were discovered, not for the order in which they participate in the photosynthetic process. The light reactions actually begin at photosystem 2. The first thing that happens is that photosystem 2 receives photons, or light energy. This light energy is transferred to a chlorophyll reaction center, causing electrons in the reaction center to become energized. These electrons become so energized that they escape photosystem 2 and move to a nearby electron acceptor molecule located in the electron transport chain. Meanwhile, to replace the electrons leaving photosystem 2, water is split, releasing oxygen, 2 hydrogen ions, and 2 electrons. The first set of electrons continues to move down the electron transport chain, releasing stored energy as it moves. This energy is used to create a hydrogen ion gradient. A protein in the electron transport chain pumps hydrogen ions from the stroma into the thylakoid space. This creates a high concentration of ions in the thylakoid space relative to the low concentration of ions in the stroma. This gradient contains a large amount of potential energy, which is used by an enzyme called ATP synthase. The hydrogen ions flow down their concentration gradient through a channel in ATP synthase, releasing energy in the process. ATP synthase uses this energy to add a phosphate to ADP, forming ATP. Let's zoom back out for a moment and return to our chloroplast. Remember from the beginning of this tutorial that the light reactions produce both ATP and NADPH. We've just seen how ATP is produced, but what about NADPH? Let's zoom back in to take a closer look. Notice these electrons over here where we left them at photosystem 1. As photosystem 1 absorbs additional light energy, the electrons again become energized, escaping photosystem 1 and moving down the second electron transport chain. electrons from the electron transport chain adjacent to photosystem 2 replace those in photosystem 1. And again, water is split to replace the electrons that have moved from photosystem 2. At the end of this electron transport chain, the energized electrons in a hydrogen molecule are used to reduce NADP to NADPH. Let's zoom back out to review. The light reactions use light energy and water to produce ATP and NADPH. Oxygen gas is released as a byproduct. Together, the ATP and NADPH formed during the light reactions are used by the Calvin cycle reactions, which are discussed more in depth in a separate tutorial. The important thing to remember from this tutorial is that plants need both light and water to survive. Without these ingredients, the light reactions would shut down, stalling photosynthesis and causing the plant to die.
Okay, so Calvin cycle is here now. It's posted. I will put the videos for all of photosynthesis in here after we're done the Calvin cycle. Okay, so we're talking part 2. Um, Calvin cycle is the official name. It's also known as the dark reactions. Okay, why do you think these are called the dark reactions? Okay, is that statement that Vedant and Emma used actually correct? Okay, so can Calvin cycle run at night? Yeah, exactly, Kirsten. So the Calvin cycle does not use light energy directly. It uses it indirectly because it still needs ATP and NADPH. And the plant can only generate that at during daylight hours. Okay? So plants use photosynthesis only during daylight. When do plants do cellular respiration? All the time. Yeah. So don't make the common mistake of saying plants do photosynthesis during day and cell respiration at night. That's not true. Plants do cell respiration all the time. Okay? Be clear on that. That's probably the most common mistake I see. Okay? Okay. So, Calvin cycle. Oops, wrong Calvin. I don't know if this joke still works because I don't know if you guys know Calvin and Hobbes or not anymore. Yes, okay. I don't know how much longer it'll last though. Okay, so this is Calvin. Okay, so he's the one who in the 1950s figured out the entire Calvin cycle, and I'm giving you an abbreviated or a simplified version, not the version that he discovered. Okay. Um, Pity the man. Any idea what his first name is? Which is why I said pity the man. No, Calvin is his last name. His first name is Melvin. So his parents named him Melvin Kelvin. Yeah. Not as bad as a kid I grew up with who was named Jack Frost. Okay. So remember what it means to be a plant. Okay. Plants need to make all organic molecules that they need for growth. So they have to make nucleic acids, carbohydrates, proteins, and fats. They have to make all four. Okay. Unlike us, where we can obtain that in our nutrients, plants can't obtain it. They have to make it. Okay. They also need to store chemical energy, ATP, produced from the light reactions, right? Because ATP is no good as a long-term energy storage molecule, so you got to convert it to something else. Okay. So a more stable form, okay, that can easily be moved around a plant. Okay. And then later used when the plant needs it, which is what this reaction is basically showing you, right? Carbon dioxide and water and energy gives you glucose and oxygen. Okay, so the light reactions, okay, converted that solar energy from the sunlight into the chemical energy that we have in ATP, which is phi, and in ADPH. Now, what can we do? Well, that's basically energy and reducing power, right? So now we can build stuff. So this is the synthesis part of photosynthesis, okay? Because we can use these two to drive synthetic synthesis reactions, right? Condensation reactions. Okay, excuse me. How is that helpful? We want to make glucose, okay, which is a synthesis procedure. How, from what, and what raw materials are available? Well, we have CO2 in the air. We know plants can grab that through their leaves. We can then reduce that carbon dioxide back to carbohydrates using the reducing power of NADPH. Okay, this process of turning carbon dioxide into carbohydrates is called carbon fixation. Okay, in this word case, the word to fix does not mean to repair. What do you think to fix something means in this context? Yeah, to attach. Okay, so if you're driving in a car and your wheel falls off, okay, you're going to fix that wheel back onto the car. You're going to reattach it, right? So fix not can also mean to attach. Okay, and then we're going to use NADPH to reduce the carbon to carbohydrates. So we're going to use ATP to fix it and NADPH to reduce it. Okay, so we're going from CO2 to glucose. Okay, carbon dioxide, as you know, has very little chemical energy in it. Okay, because it's fully oxidized. Okay, whereas glucose has a lot of chemical energy, okay? Because it's highly reduced, right? It has a lot of hydrogens attached to it. Synthesis, as we've already discussed, is endergonic. It requires energy, okay? So we have to get a lot of energy to do this, okay? So the reduction of carbon dioxide to glucose proceeds in a lot of little uphill steps. It's effectively the reverse of cellular respiration, where we're going the other way, proceeding in many small downhill steps. Okay, catalyzed by an enzyme. Okay, and using the energy in ATP and NADPH. So that's how we basically do this. Okay, so going from the light reactions to the Calvin cycle. Okay, the Calvin cycle occurs in the stroma of the chloroplast, so it does not occur in the membranes. Okay, it happens in the fluid-filled interior of the chloroplast, not in the thylakoid membranes, not in the space of the thylakoid membranes, but out here in the main fluid of the chloroplast. Okay, you need to get the products of the light reaction here to drive this cycle to run. Okay, so you need those two products. Okay, yeah, go ahead. Careful. Okay. Um, actually, this slide didn't translate too well, so I'm going to show it to you. Over here, I think. Yeah, okay, hang on. I got to move you guys over here. Okay, I got to power that. Present a window. Share. Okay, so this cycle starts, okay, with a 5-carbon molecule called RUBP. You don't have to know the full name. If you do, great, but I'm not going to worry about it. So the 5-carbon compound that we need is something called ribulose biphosphate. Okay. You're also going to have coming in at the same time a CO2 molecule. Okay. So we've got 3 5-carbon molecules here, which means we need 3 CO2s coming in here. Okay. The enzyme that's going to combine these two molecules together is called Rubisco. You need to know the name of this enzyme. The short form Rubisco is fine. You don't need to know ribulose biphosphate carboxylase. But the enzyme that combines carbon dioxide to this ribulose biphosphate is Rubisco. Okay? That's going to give you a 6-carbon molecule here. Okay. The problem with the 6-carbon molecule is that this 6-carbon molecule is completely unstable. Okay. And because it's unstable, it's going to immediately split into 2 3-carbon molecules. Okay. So this step, where the carbon dioxide is attached to the RUBP, is carbon fixation, stage 1. Okay. The 6-carbon molecule, which because it's so unstable, it doesn't even get a name. Okay. Is going to break into 2 3-carbon molecules. And this 3-carbon molecule is called phosphoglycerate, PGA. Okay? Excuse me, I gotta close the door. Now, you're now going to use, because that way the math works out, we can compare it to Krebs. I could do it just once, but then we have to run the cycle 6 times to make 1 glucose. Okay, so we're going to use 6 ATPs, okay, and they're going to phosphorylate, okay, the 3-carbon molecules, okay. I'm not going to show you the phosphorylation step. You're then going to take 6 NADPHs and you're going to reduce this 3-carbon molecule to NAD, okay? So you're going to end up with NADP. What's going to happen to the NADP and the ADPs? What are they going to do? Yeah, they're going to go back to the light reaction to generate more ATP and NADPH, okay? Now, what's happened here, okay, we're going to a new 3-carbon molecule. This is the reduction step, right? Because we've reduced this 3-carbon molecule. What's actually happened going from here to here, we've taken these double bonds and we've added hydrogens to them. So we've reduced this molecule to this one effectively. Got rid of the double bonds that were in it. Which is why I changed colors from blue to yellow. Okay. Now, at this point, okay, we got to get back to RUBP. How many carbons did we start with here? 15. How many carbons do we have here? 18. So that means we got to get rid of 3 carbons, right? Because if we tried to get this back to here, we'd have three extra carbons. We want to have 15 total, not 18. So what happens is one of the 3 carbon molecules is removed. Okay, this molecule is G3P, which is the same molecule you found in glycolysis. This one G3P is removed, okay, and is used to make glucose. So we have half of a glucose molecule right here, which is why we need to run this sequence twice to get a full glucose molecule. This is what I meant by we have to run this to get the math to work out to the dant earlier. Okay, that glucose molecule can now go on to make starch or sucrose or cellulose or proteins or lipids or nucleic acids. Okay, so there's your synthesis bit. The remaining 5 carbon molecules are going to use another 3 ATP to get back to RUBP. Okay, oops, let me go back. Now, so I'll go through all of this to get back to where we were. Okay, so we're back to the RUBP. Okay, okay, remember the G3P, right? We had it in glycolysis. Okay, what we're doing in photosynthesis is from Calvin cycle, we're basically coming here and we're running up. Okay, so we're running the first half of glycolysis backwards effectively. In general, that's what we're doing. It's not the exact same chemical steps run backwards, But from the point of view of making glucose, that's what we're basically doing. Okay? So we're going to G3P, and then, to quote Tolstoy, we're going to G3P and beyond. Okay? So G3P is the end product of the Calvin cycle. Okay? It is a very energy-rich 3-carbon sugar. Okay? So this process is called that C3 photosynthesis, because the first stable product that we make before we make glucose is a 3-carbon glyceraldehyde 3P. Okay? So G3P is a really important intermediate, because from GP3 you can make glucose, and then from glucose you can make carbohydrates. From G3P you can make lipids, you make amino acids, and you can make nucleic acids. So G3P is the molecule that allows the plant to make all of the other macromolecules that it needs, which is why Calvin cycle is so critical. Okay, so Rubisco, right, the enzyme that fixes carbon dioxide, okay, is probably the most important enzyme in the world. Why? Not only used for plants, right? Animals, heterotrophs, couldn't exist without the existence of Rubisco. Okay? Could plants live without consumers? Yes. Can consumers live without producers? Right? This is why it's so important. Okay? So this molecule literally makes life out of air. Okay? It's most definitely the most abundant enzyme on this planet, right? Because it's present in huge amounts in every single plant on this planet. Okay, let's look at the accounting. It is complicated, and I'm giving you the simple version. Okay, so it takes 3 turns of the Calvin cycle to make 1 G3P, because every turn of the Calvin cycle only captures 1 carbon from the air. Okay, so 3 carbon dioxides gives you 1 G3P. So it takes 6 turns of the Calvin cycle to make a glucose, because there's 6 carbons. 6 CO2s gives you 1 glucose. That needs 18 ATPs and 12 NADPHs. Okay. Any ATP left over from the light reaction then can be used by other processes inside the cell. Okay. So that's how this is accounted for. Okay. So summary. Light reactions. I'm going to summarize both and then combine it. Produced ATP and NADPH. Consumed water and produced oxygen as a byproduct. Calvin cycle consumes carbon dioxide, produces G3P, regenerated the ADP, and regenerated NADP+. Okay. So water comes in, ATP comes out, NADPH comes out, Oxygen leaves. Okay. Calvin cycle is going to use the NADPH and ATP plus CO2 from the air to make sugars. Make regenerating ADP and NADP plus, which go back to the light reactions. Okay, so builds sugars, okay, uses ATP and NADPH, okay, and recycles ADP and NADP, okay, back to the light reactions to make more ATP and NADPH. So putting it all together, sunlight, water makes ATP and NADPH, releasing oxygen. Then carbon dioxide comes in, building sugars, using ATP and NADPH, regenerating ADP and NADP plus. Okay, so plants make both energy, ATP and NADPH, and sugars. Okay, so if we look at the cycle, okay, don't worry about these funky arrows. Photosynthesis, they didn't translate it over from Photoshop, not Photoshop, from PowerPoint very well. Okay, is going to take carbon dioxide and water and light energy and make glucose and oxygen. Okay, why is this equation a little bit of a lie? The way it's currently written, why is it a lie? Not only is it not a direct equation, what's the lie? Yeah, you're not combining CO2 and water to make glucose. The water is used in the light reaction, CO2 is used in the Calvin cycle. So it makes a better story, which is why we present it this way, but this isn't actually true. Okay, so that's plants. They're going to use, so the glucose and water is going to be used by us in cellular respiration to generate ATP, make CO2 and water, which will cycle back to photosynthesis. Okay, so that is the great circle that we talk about. Okay, summary. Where did the carbon dioxide come from? Okay, the air and ultimately cellular respiration, waste product. Where did the CO2 go to? Okay, G3P and carbohydrates. Where did the water come from? Okay, cellular respiration again. Okay, where's the water go? They're split, yeah, they go into the light reactions. Where did the energy come from to run the process? Sun. What's the energy used for? Which is then used for? Because it's all of photosynthesis, right? Yeah, it's used to make sugar. What will the sugar then be used for? Structure, cellular respiration, synthesis, yeah. Where did the O2 come from? Okay, it came from the splitting of water. Where will the O2 go? That ends up in the air. Okay, what else is involved not listed in this equation? ATP and ADPH. Okay, so on a global scale, photosynthesis is the most important process for continuation of life on this planet. Okay, every year photosynthesis captures about 121 billion tons of CO2. Okay. So if you think about that, the impact that humans have causing CO2 levels still to go up, we're dumping a huge amount of CO2 into the air then. Okay. 160 billion tons of carbs are made every year by plants. Okay. All heterotrophs then depend on plants as a food source. Okay. And from a poetry point, all the solid material of every plant was built by sunlight out of thin air. All the solid material of every animal was built from plants. So all the people, the cats, the dogs, elephants are really particles of air woven together by strands of sunlight. Okay. All right. So it's 9:54. We got a wee bit of time, so let's take a bit of a longer break today. We'll take it for 15 minutes. So I'll see you guys at 9:10. Sure. Which part? In the Kelvin cycle or what the term fixing means? So that's basically this. Rubisco grabbing the carbon, attaching it to RUBP, the carbon's been fixed. Let me present that part. Right? You take 3 RUBPs, attach, each carbon gets attached to one of the 5 carbon molecules by the enzyme Rubisco. That's carbon fixing. Yes. Okay. Okay, I'm just adding some videos. So let's say crash course on photosynthesis. We also want Bozeman science photosynthesis. Control C. Add YouTube. And probably an Amoeba Sisters. Synthesis. Is that the updated one? Yeah, okay, that is the one. Okay. Copy. Add YouTube. Add. Save. Okay, I think I'm going to show you guys the crash course one.
Photosynthesis. It is not some kind of abstract scientific thing. You would be dead without plants and their magical, nay, scientific ability to convert sunlight, carbon dioxide, and water into glucose and pure, delicious oxygen. This happens exclusively through photosynthesis, a process that was developed 450 million years ago and actually rather sucks. It's complicated, inefficient, and confusing, but you are committed to having a better, deeper understanding of our world, or more probably, you'd like to do well on your test, so let's delve. There are two sorts of reactions in photosynthesis, the light-dependent reactions and the light-independent reactions, and you've probably already figured out the difference between those two, so that's nice. The light-independent reactions are called the Calvin cycle. Yes, that Calvin cycle. Photosynthesis is basically respiration in reverse, and we've already covered respiration, so maybe you should just go watch that video backwards. Or you could keep watching this one. Either way, I've already talked about what photosynthesis needs in order to work: water, carbon dioxide, and sunlight. So how do they get those things? First, water. Let's assume that we're talking about a vascular plant here. That's the kind of plant that has pipe-like tissues that conduct water, minerals, and other materials to different parts of the plant. These are like trees and grasses and flowering plants. In this case, the roots of the plants absorb the water and bring it to the leaves through tissues called xylem. Carbon dioxide gets in and oxygen gets out through tiny pores in the leaves called stomata. It's actually surprisingly important that plants keep oxygen levels low inside of their leaves for reasons that we will get into later. And finally, individual photons from the sun are absorbed in the plant by a pigment called chlorophyll. Alright, you remember plant cells? If not, you could go watch the video where we spend the whole time talking about plant cells. One thing that plant cells have that animal cells don't: plastids. And what is the most important plastid? The chloroplast, which is not, as it is sometimes portrayed, just a big fat sack of chlorophyll. It's got complicated internal structure. Now, the chlorophyll is stashed in membranous sacs called thylakoids, and the thylakoids are stacked into grana. Inside the thylakoid is the lumen, and outside of the thylakoid, but still inside of the chloroplast, is the stroma. The thylakoid membranes are phospholipid bilayers, which, if you remember, means that they are really good at maintaining concentration gradients of ions and proteins and other things. This means keeping a concentration higher on one side than the other of the membrane. You're going to need to know all of these things, I'm sorry. Now that we've taken our little tour of the chloroplast, it's time to get down to the actual chemistry. First thing that happens, a photon created by the fusion reactions of our sun is about to end its 93 million mile journey by slapping into a molecule of chlorophyll. This kicks off stage one, the light-dependent reactions, proving that yes, nearly all life on our planet is fusion powered. When chlorophyll gets hit by that photon, an electron absorbs that energy and gets excited. This is the technical term for electrons gaining energy and not having anywhere to put it, and when it's done by a photon, it's called photoexcitation. But let's just imagine, for the moment anyway, that every photon is whatever dreamy young man 12-year-old girls are currently obsessed with, and electrons are 12-year-old girls. The trick now, and the entire trick of photosynthesis, is to convert the energy of those electrons into something that the plant can use. We are literally going to be spending the entire rest of the video talking about that. I hope that that's okay with you. That first chlorophyll is not on its own here. It's part of an insanely complicated complex of proteins and lipids and other molecules called photosystem II that contains at least 99 different chemicals, including over 30 individual chlorophyll molecules. This is the first of 4 protein complexes that plants need for the light-dependent reactions. And if you think it's complicated that we call the first complex photosystem II instead of photosystem I, then you're welcome to call it by its full name, which is plastoquinone oxoreductase. Oh, no, you don't want to call it that? Right then, photosystem II. Or, if you want to be brief, PS2. PS2 and indeed all of the protein complexes in the light-dependent reactions straddle the membrane of the thylakoids in the chloroplasts. Now that excited electron is going to go on a journey designed to extract all of its new energy and convert that energy into useful stuff. This is called the electron transport chain, in which energized electrons lose their energy in a series of reactions that capture the energy necessary to keep life living. So PS2's chlorophyll now has this electron that is so excited that when a special protein designed specifically for stealing electrons shows up, the electron actually leaps off of the chlorophyll molecule onto the protein, which we call a mobile electron carrier because it's a mobile electron carrier. The chlorophyll then freaks out like a mother who has just had her 12-year-old daughter abducted by a teen idol and is like, what do I do to fix this problem? And then it, in cooperation with the rest of photosystem II, does something so amazing and important that I can barely believe that it keeps happening every day. It splits that ultra-stable molecule, H2O, stealing one of its electrons to replenish the one it lost. The byproducts of this water splitting, hydrogen ions, which are just single protons, and oxygen. Sweet, sweet oxygen. This reaction, my friends, is the reason that we can breathe. Brief interjection. Next time someone says that they don't like it when there are chemicals in their food, please remind them that all life is made of chemicals, and would they please stop pretending that the word chemical is somehow a synonym for carcinogen? Because, think about how chlorophyll feels when you say that. It spends all of its time and energy creating the air we breathe, and we're like, ew, chemicals are so gross. Now remember, all energized electrons from PS2 have been picked up by electron carriers and now are being transported to the second protein complex, the cytochrome complex. This little guy does 2 things. 1, it serves as an intermediary between PS2 and PS1, and 2, it uses a little bit of that energy from the electron to pump another proton into the thylakoid. So the thylakoid's starting to fill up with protons. We've created some by splitting water, and we moved one in using the cytochrome complex, but why are we doing this? Well, basically what we're doing is charging the thylakoid like a battery. By pumping the thylakoid full of protons, we're creating a concentration gradient. The protons then naturally want to get the heck away from each other, and so they push their way through an enzyme straddling the thylakoid membrane called ATP synthase. And that enzyme uses that energy to pack an inorganic phosphate onto ADP, making ATP the big daddy of cellular energy. All of this moving along the electron transport chain requires energy, and as you might expect, electrons are entering lower and lower energy states as we move along. This makes sense when you think about it. It's been a long while since those photons zapped us. We've been pumping hydrogen ions to create ATP, it's splitting water and jumping onto different molecules. I'm tired just talking about it. Luckily, as 450 million years of evolution would have it, our electron is now about to get re-energized upon delivery to photosystem 1. So PS1 is a similar mix of proteins and chlorophyll molecules that we saw in PS2, but with some different products. After a couple of photons re-excite a couple of electrons, the electrons pop off and hitch a ride onto another electron carrier. This time, all of that energy will be used to help make NADPH, which, like ATP, exists solely to carry energy around. Here, yet another enzyme helps combine 2 electrons and 1 hydrogen ion with a little something called NADP+. As you may recall from our recent talk about respiration, there are these sort of distant cousins of B vitamins that are crucial to energy conversion. In photosynthesis, it's NADP+, and when it takes on those 2 electrons and 1 hydrogen ion, it becomes NADPH. So what we're left with now, after the light-dependent reactions, is chemical energy in the form of ATPs and NADPHs. And also, of course, we should not forget the most useful, useless byproduct in the history of useless byproducts, oxygen. If anybody needs a potty break, now would be a good time, or if you want to go rewatch that rather long and complicated bit about light-dependent reactions, go ahead and do that. It's not simple, and it's not going to get any simpler from here. Because now we are moving along to the Calvin cycle. The Calvin cycle is sometimes called the dark reactions, which is kind of a misnomer because they generally don't occur in the dark. They occur in the day along with the rest of the reactions. But they don't require energy from photons, so it's more proper to say light-independent reactions. Or if you're feeling non-descriptive, just say stage 2. Stage 2 is all about using the energy from those ATPs and NADPHs that we created in stage 1 to produce something that's actually useful for the plant. The Calvin cycle begins in the stroma, or the empty space inside of the chloroplast, if you remember correctly, and this phase is called carbon fixation because, yeah, we're about to fix a CO2 molecule onto our starting point, ribulose bisphosphate, or RUBP, which is always around in the chloroplast because not only is it the starting point of the Calvin cycle, it's also the end point, which is why it's a cycle. CO2 is fixed to RUBP with the help of an enzyme called ribulose 1,5-bisphosphate carboxylase oxidase, which we generally shorten to Ribisco. I'm in the chair again. Excellent. This time for a biography of Ribisco. Once upon a time, a one-celled organism was like, man, I need more carbon so I can make more little me's so I can take over the whole world. Luckily for that little organism, there was a lot of CO2 in the atmosphere, and so it evolved an enzyme that could suck up that CO2 and convert inorganic carbon into organic carbon. This enzyme was called Ribisco, and it wasn't particularly good at its job, but it was a heck of a lot better than just hoping to run into some chemically formed organic carbon. So the organism just made a ton of it to make up for how bad it was. Not only did the little plant stick with it, it took over the entire planet, rapidly becoming the dominant form of life. Slowly, through other reactions known as the light-dependent reactions, plants increased the amount of oxygen in the atmosphere. Ribisco, having been designed in a world with tiny amounts of oxygen in the atmosphere, started getting confused. As often as half of the time, Ribisco started slicing ribulose bisphosphate with oxygen instead of CO2, creating a toxic byproduct that plants had to deal with in creative and specialized ways. This byproduct, called phosphoglycolate, is believed to tinker with some enzyme functions, including some involved in the Calvin cycle. So plants have to make other enzymes that break it down into an amino acid, glycine, and some compounds that are actually useful to the Calvin cycle. But plants had already sort of gone all in on the Ribisco strategy, and to this day, they have to produce huge amounts of it. Scientists estimate that at any given time there are about 40 billion tons of Ribisco on the planet. And plants just deal with that toxic byproduct. Another example, my friends, of unintelligent design. Back to the cycle. So ribulose bisphosphate gets a CO2 slammed onto it, and then immediately the whole thing gets crazy unstable. The only way to regain stability is for this new 6-carbon chain to break apart, creating 2 molecules of 3-phosphoglycerate. And these are the first stable products of the Calvin cycle. For reasons that will become clear in a moment, we're actually going to do this to 3 molecules of RUBP. Now we enter the second phase, reduction. Here, we need some energy, so some ATP slams a phosphate group onto the 3-phosphoglycerate, and then NADPH pops some electrons on, and voila! We have 2 molecules of glyceraldehyde 3-phosphate, or G3P. This is a high-energy 3-carbon compound that plants can convert into pretty much any carbohydrate, like glucose for short-term energy storage, cellulose for structure, starch for long-term storage. And because of this, G3P is considered the ultimate product of photosynthesis. However, unfortunately, this is not the end. We need 5 G3Ps to regenerate the 3 RUBPs that we started with. We also need 9 molecules of ATP and 6 molecules of an ADPH. So with all these chemical reactions, all of this chemical energy, we can convert 3 RUBPs into 6 G3Ps, but only one of those G3Ps gets to leave the cycle. The other G3Ps, of course, being needed to regenerate the original 3 ribulose bisphosphates. That regeneration is the last phase of the Calvin cycle. And that is how plants turn sunlight, water, and carbon dioxide into every living thing you've ever played with, climbed on, loved, hated, or eaten. Not bad, plants. And I hope you understand. If you don't, not only do we have some selected references below that you can check out, but of course, you can go rewatch anything that you didn't get. And hopefully, upon review, will make a little bit more sense. Thank you for watching. If you have questions, please leave them down in the comments below.
Okay, so that's all of photosynthesis. I would say this is actually a pretty good one to look at if you're getting confused. You could probably skip the biography, but otherwise, it's a pretty good version. Why is this not going back? Okay, classroom. Okay, here we go. B block, classwork. Okay, the next thing we have to look at, and I will add it now, is something called and C4 pathways. Uh, Burnett methods. Add Google Drive. Okay. This is actually the last PowerPoint for plants. Should be. Let me just double check that. Calvin cycle can. We're not doing factors that affected photosynthesis. So yeah, we're basically at the end of the unit. Let me just double check something. Biology, education, no, not curling, curriculum. 11, 12. Market changes. Yeah, okay, so with this, we're done. So what'll probably happen, I'll finish the lectures today. We'll probably do a gizmo tomorrow, which basically means for this unit, it will have been two labs for this unit rather than a lab and a paper. Okay. So I'm not going to give you guys another paper to do. I'm just going to give you. I'm going to give you a second. Case study. Just make sure that it's there. want to be sure. Find gizmo. Photosynthesis. There's a stem case. Yeah, okay. So yes, we'll be doing a photosynthesis instead. Probably about the same time that we have your test on Tuesday. I mean, theoretically, you could do it tomorrow, right? In a 1-day period. Some of you will have 2 gizmos then to complete because I'd say 6 of you haven't completed the first one. But realistically, this shouldn't take you more than an hour to do. So I'm not sure why for some of you it's taking longer when I give you a full period, but whatever. Um, just get them done because we are fairly rapidly running out of time. What are we at? Today is the 18th. We've got 1, 2, 3, 4 weeks and 3 days, and we're done the course, right? So we don't have a heck of a lot of time. Because we're finished on the 18th, right? The days after the 19th, 20th, the 21 and 22nd are basically going to be support days, catch-up days. So if you have stuff you're not done, you have those days to, actually not even. You'll have the 21st to catch up. The 22nd, if we can, if you have stuff to hand in still, you can hand it in. I will try to change your marks, but your marks are due 8:30 a.m. on the June 22nd, right? Because they get uploaded to OWAC on the 23rd. So dates are going to come flying by really fast now, especially with the good weather. And you guys are going to get more and more senioritis. I know that. Be aware of that because deadlines are going to be fast. Okay, let's take a look at this. Come on, load up. Okay. So we're looking at variations on the theme. We're looking at what happens when things don't go quite the way they're supposed to for photosynthesis. Okay. Now, what do I mean by that? Okay. Remember what plants need in order to do photosynthesis. Okay. They need light coming in from the sun. They need water coming in from the ground. And then for the Kelvin cycle, they need CO2, which comes from the air. Right? So the structures that plants have evolved to meet these needs: leaves and roots. Okay? That's pretty basic. Okay, it's stomates in the leaves. Okay, stem to move water and sugars up and down. Okay, let's look at we need to, remember I said you should know the parts of a leaf. That's because it comes into play now. Okay, so you've got the cuticle, which is the waxy layer on the outside. Then you've got the upper and the lower epidermis. And then you've got 2 layers. One is called the palisade layer, and the other is called the spongy layer, which has air gaps. And then you've got the vascular bundle, which is basically xylem and phloem cells. Okay. Now, water, right, um, is going to evaporate into the airspace. And then if the stomates are open, they're going to leave the interior of the leaf via transpiration. Okay. So water is going to continuously flow out, which is problematic because it means plants start to dehydrate. Okay. I just need to check on something. Hang on. There we go. Okay, so plants are dehydrating. Now, oxygen at the same time, right, is also going to leave the cell, the interior of the leaf, just by straight gas exchange because the stomates are open. Okay. CO2, on the other hand, is going to come in from the atmosphere into the air spaces because it needs it for photosynthesis. So again, that's just straight gas exchange. Okay. So these are the three products that are basically moving in and out of the plant. Okay. So you've got gas exchange of carbon dioxide and oxygen, and transpiration causing the removal of water. Okay. So that's going to have water up here is going to evaporate from the xylem, move from the xylem into the bundle sheath cell, and then out to the airspace, and then out of the cell. Carbon dioxide is going to be moving through as sap, going to the rest of the plant. Okay, so that's the vascular bundle. Okay, now the problem for plants is that they have to control the loss of water from the leaves. On really, really hot or really, really dry days, okay, in order to conserve water, to prevent that water from being transpired out of the leaf, the stomates are going to close up, okay, or not completely, but they're going to get really, really freaking small, okay, right? So the guard cells are going to close up, okay? So that's an adaptation that plants have to living on land because they need to keep the water. But this immediately starts causing problems for the plant because when the stomates close, what's going to happen to the movement of water out of the leaf? Go up or go down? Okay, there'll be a little bit of it because it's not a complete closure, but there is a reduction of water loss from the plant, which is what we want. Okay, but what's going to happen to the uptake of CO2? No, this is the same unit, right? So carbon dioxide is going to come in more slowly. And what's going to happen to the amount of oxygen inside the leaf? Right. So the next unit is genetics. So we're still in plants metabolism, Abby. So you're going to have a drop in CO2 levels in the leaf, and you're going to have a buildup of oxygen. Okay. Closed stomates lead up to a buildup of oxygen because the light reactions are going to keep going. Because it's just a pore. So if it's a hole, things are just going to go in and out. But the opening and closing of stomates is controlled by water content. Right? The air holes are never specific. No, because they just lead to a air gap. Right? I mean, you breathe in and out through your lungs and nose. You don't have one tube for oxygen and another tube for CO2. Right? That's over engineering. Okay. CO2 is depleted because the Calvin cycle keeps running. Okay. So that means you now have problems with the Calvin cycle because you're not getting enough carbon dioxide. Okay. So here at the stomates, things are basically, it's good for water, okay, because you're not losing water, but now O2 and CO2 can't as well, okay? With Rubisco, it's basically a following. Yeah, it looks like it. Okay, there's one molecule. What do you notice about the shape of this molecule? Okay, CO2. What's its structure? How would you describe its structure? Okay, it's linear. How would you describe this molecule? Exactly the same, right? It's a linear molecule. Okay. In our atmosphere today, which one is more abundant? Yeah. Okay. What's the percentage of O2 in our air? It's about 20%, 19 point something. What's the percentage of carbon dioxide in the air? Lower, lower, lower. It's about 0.03. Okay. It's pretty low. Okay. Um, actually, 0.3 might be right. I can't remember. CO2 content. I think it's 0.03. CO2. At any rate, um, so they're both very linear structures. Okay, so normally Rubisco is supposed to bind this molecule because it's the carbon fixing enzyme in photosynthesis. Okay, so when Rubisco binds CO2 in its active site, okay, you're going to reduce the RUBP and you're going to build sugar. The problem is it possible for this to fit the active site of Rubisco as well? Yeah, because they're not that different in terms of shape. And if oxygen concentrations are high, Rubisco binds O2, not CO2. Okay? Considering this is 20% and this is about 0.03%, maybe 3.3%, the likelihood of Rubisco binding this rather than this is really, really high. And when Rubisco binds O2, okay, so O2 is a competitive inhibitor of Rubisco, okay, it oxidizes RUBP and it breaks the sugar down, okay, which is problematic. Biology, let me just check something. I want to check that percentage again. Yeah, 0.03%. It is 0.03. So it is really low. Okay, so the problem is if Rubisco grabs this, it starts breaking down carbohydrates. How big of a, and this process is called photorespiration. Okay. The problem with photorespiration, okay, when Kelvin cycle is running normally, this is what we usually see. Carbon dioxide gets fixed, becomes an unstable intermediate, and then it runs through the system to make G3P. This is what's supposed to happen. Okay. Okay. And that's why we have C3 plants. The problem in a C3 plant is that if oxygen levels are high, O2 combines with Rubisco and a 5-carbon product to make a 3-carbon and 2-carbon molecule. The 2 carbon molecule goes to the mitochondria where it's lost that CO2 but does not make ATP. So there's energy loss here. The 3 carbon molecule tries to complete the cycle, and what ends up happening here is that you actually have to take glucose and make another RUBP to make up for the 2 carbons that were lost. Generates no ATP energy. For a plant, that's bad. Okay? So the impact of photorespiration, hold me for time. Okay. We're oxidizing RUBP. Okay. So we're short-circuiting the Calvin cycle. Okay. We lose carbons to CO2. Okay. And here's the bad part. C3 plants on a really hot day can lose half of their carbons that were fixed in the Calvin cycle back to CO2. So basically, they're taking 2 steps forward and 1 step back every time. Okay, so it reduces the products of photosynthesis. Okay, there's no energy produced. Okay, no food is produced. Okay, so if you could get rid of or reduce photorespiration, plants would become 50% more efficient. Okay, so there's a lot of selection pressure to evolve a different carbon fixing method. We'll talk about that tomorrow, and then we'll do the gizmo. Okay, but what I want you to think about. Okay, let me just stop presenting. So I want you to think about this question. I want you to think about and try to figure out, why is there a C3 problem? Why does Rubisco have the ability to use CO2 or O2? Why isn't its active site more specific? See if you can come up with an answer for tomorrow. Okay, that's your homework. And see if you can do it without Googling and without looking up the rest of the PowerPoints. Okay, so I'll see you guys. Yeah, it's 10:49, so I'll see you guys tomorrow. Yeah, a specific reason as to why this occurs. And see if you can do it with